with a haploid cell, you're going to end up with a haploid cell. If you start out with a diploid cell, you're going to end up with a diploid cell. So whatever you start out with, you're going to end up with. Okay, so here's the big picture of mitosis. This is a really fantastic drawing on my part. Those chromosomes are really looking identical. What do you think? Not so much? Okay, whatever. What do you think? Arrow, square. I'm digging the star. I'm going to stick with the star. Okay, there it is. All right, so this is the big picture. And remember, you need to go back and remind yourself what prophase is, what metaphase is, what anaphase is, and what telophase is. And because that's really important. But those are big details. And this is just the quick review for MCAS, for our state test. So we're starting out with a cell. Okay, in this case, it's got a red chromosome and a blue, and then it goes through the S phase. It goes through DNA replication. So we end up doubled. Then we're going to go through mitosis. We're going to go through prophase, metaphase, anaphase, and telophase. So the chromosomes are going to condense. Then they're going to line up across the equator. Then they're going to start to spread apart. And then over here, we're going to hit telophase, where they're going to be at the far ends. And then we're going to get to cytokinesis, where they break apart, and we end up with two identical daughter cells. So they are identical to what this cell looked like before it went through DNA replication. And that's really important to keep in mind. So practicing drawing something like this might help you keep it straight, because if you can't remember this super duper important piece that it goes through the DNA replication, you're going to mess it up. So for example, if it was a human cell, it would have 46 chromosomes at the beginning. It would go through DNA replication and have 92, and at the end each cell would have 46. So hopefully that makes some sense. Okay, so meiosis, okay, totally different. We're making 4 unique haploid cells. We're making sex cells. Oops. Okay. Ooh, I ended up with a star with an outline. I don't even know how to do that, but that's cool. All right, so remember that it's similar in the fact that cell division is similar, but we're getting a very different process, and we're ending up with unique cells. And I forgot to put crossing over in here, but I'll write that in. So anyway, our germ cell is right here. That's our first cell, is a germ cell. That's that one that we start out with, which, like all our other cells, has 46 chromosomes. Okay, and it goes through, oops, I didn't mean to make that a unit. Oops, I need another star now. Okay. Okay, so now we have 92. They're going to go through PMAT once, Paul makes taco, meiosis 1. And this is where crossing over occurs. So homologous chromosomes, chromosomes that have the genes for the similar things, are going to line up, and then they're going to exchange their genes. So crossing over is going to occur in prophase 1. And I don't think I have anywhere to write that, to have that work out for me. Can I write that in here? I can't really write that small. Crossing over happens in P1. So crossing over happens in prophase 1, and that's where genes, we jokingly call it changing genes, like you're changing your genes like your clothes, but that's, so if that helps you remember it, like you're changing genes, that you're switching something, that's a good way to remember it. So it happens in meiosis 1, it happens in prophase 1, that homologous chromosomes change their genes. Okay? And so after we do our first PMAT, we end up with 46. Okay, And then we're going to have these two split again. So now we have 4 unique cells that are going to go on to be further developed to become sperm. At this point, they're a little underdeveloped and they're not eggs yet either. And actually, only one of them is going to go on to become an egg, but that's another story. But anyway, so at this point, they're just at the beginning stage of becoming that. But it's a unique gamete with a unique bunch of chromosomes because you are the product of half your mother's genes and half your father's genes, a unique mixture. And so, say this 23 right here becomes a sperm, well, that's already a unique mixture of the genes from, if this is going to be sperm, from this father. So that it's a unique mixture of the father's genes to pass on. Okay, and I'm going to add one more to talk about fertilization. Okay, so fertilization, then the last bit of this picture is when we have, say we have our sperm once it's been developed, okay, with its 23 chromosomes, and I guess I'll pick a pink egg there. We have our pink egg with 23, and when these two meet, these two gametes, when they meet, we call that fertilization, and they're going to form one cell that is 46 chromosomes, which is going to be the first cell of a human, and that is called a zygote. And so we all begin as one cell that is a zygote. So that is the end of product of forming two 23 chromosome unique cells from meiosis and getting them together to form a zygote. And that's the end of this little quick review. So please remember to go back and review the phases in detail. Prophase, metaphase, anaphase, and telophase for both mitosis and meiosis. And you can see some great ones online. But looking at pictures seem better than me explaining them out loud, because if you look at the pictures, you'll know them really quick. All right, that's it for this.